In the next few minutes, I will explain why a differential amplifier is useful for acquiring voltage measurements at the surface of the human body. Voltage measurements at the surface of the body are very small, as little as 10 microvolts in some sensory nerves, and this is very small when compared to the mains pickup of around 10 volts. We therefore need to amplify the signal and cancel the noise so that it can be processed and interpreted. Differential amplifiers are used to do this. This diagram gives an overview of how a basic operational amplifier works. There are two input voltages with a differential voltage between them. This voltage is multiplied by the gain of the amplifier, A, to give the output voltage. There are some specific requirements of the op-amp to provide the best biopotential measurements. The first is that there is a high input impedance. This is so as not to load the signal being amplified and therefore reduce the distortion that electronic loading on the electrodes can cause to the signal. It also minimises the motion artifacts because the amplifier will be less sensitive to fluctuations in electric impedance that result from movement of the tissue. There should also be a low output impedance in order to maintain the maximum signal output. Finally, the common mode rejection ratio should be high. This is important when the voltage fluctuation is small compared to a large voltage offset. The common mode voltage is a voltage that appears at the input terminals and is separate from the signals we are trying to measure. It is much bigger than the very small biopotential signal that we are trying to measure and will also be amplified along with this biopotential signal. It is therefore important that it is rejected. We need an amplifier with a high common mode rejection ratio to reject the signal so that the output voltage is the pure biopotential amplified. A differential amplifier can fulfill these three requirements. One type of differential amplifier is the one op-amp differential amplifier. This setup gives a very high common mode rejection ratio. It can be higher than 10,000, but the input resistance is too low for this application. To solve this, two more op-amps are added to give a 3 op-amp differential amplifier. These two op-amps provide buffering and provide extra gain. While this setup provides the high input resistance that we need, it does not improve the common mode rejection ratio because it will amplify both the biopotential and the common mode voltage. This diagram simplifies the 3 op-amp differential amplifier we have just seen. When measuring voltages at the surface of the body, V2 is the reference electrode. The reference electrode is important because it means that, for example, to take an ECG, the output of all the leads can be measured with respect to one point. In this diagram, the 3 limb leads from the right arm, and left leg are connected to the Wilson central terminal, which is the average of the voltages at the 3 electrodes. This reference is also used for the precordial leads on the chest. This is one example of a reference electrode. Another one could be by removing the connection of a limb being measured, in this example from the right arm, so that the reference electrode is the average of the other 2 voltages. The leads are now called augmented leads. An EEG uses many electrodes on the surface of the head, and a variety of things may be used as a reference electrode. A common reference. This is when one electrode is used as a reference for all the channels, the same one each time. Average reference. This is similar to the Wilson central terminal that we have just seen. The reference is the average of all the electrodes. Bipolar. This is when the reference is formed by linking electrodes together in a specific sequence. To summarise, differential amplifiers are used because voltages at the surface of the human body are very small compared to the mains pickup voltage and need to be amplified and noise reduced. Differential amplifiers have a high input impedance, low output impedance, and a high common mode rejection ratio. The differential amplifier used to measure biopotentials consists of 3 op-amps. Reference electrodes are necessary to gain a comprehensive analysis of the voltages being measured. In ECGs and EEGs, there are numerous different ways of creating this reference electrode.